Hello, grade 12 learners. We're now going to go through meiosis. But before we can actually do meiosis, we need to just recap on mitosis. Now remember, mitosis was for growth, repair, replacement, and reproduction in unicellular organisms. In other words, reproduction when there is only one cell. There aren't different cells for reproduction like in our bodies, where we have the testes which produce sperm cells and the ovaries which are going to produce egg cells. If you only have one cell, well, guess what? That one cell has to reproduce, and it's a form of mitosis. It's called binary fission. So mitosis is for growth, repair of cells, replacement of cells when a cell can't be repaired properly, and then reproduction in unicellular organisms. Now, if we look at mitosis versus meiosis, remember it this way. When your toes grow, your shoes get too small for you. So mitosis, growth, repair, replacement. If I get a blemish on my skin, or you get a pimple, or your lips get very dry and they get chapped, how do they heal? By mitosis. If I lose a liver cell or a heart cell or a lung cell, how do I make sure that there's another cell that's identical as the cell next to it produces a new cell? So that is by mitosis. Otherwise, you'll end up with a heart cell here pulsing away on your lip, and that would be no good. So the cells, where there is a damaged cell or a cell that must be repaired or replaced, it's going to be the cells next to it that undergo mitosis. Also to grow hair, to grow nails, to replace bone cells all the time. The only cells that don't get replaced, and listen carefully, are your brain cells. These guys here, they grow in size, so your brain gets slightly bigger from birth, but you'd never ever replace brain cells. So don't throw them away by doing silly things. But that's mitosis. When my toes grow, my shoes get too small for me. Growth, repair, replacement, and reproduction in unicellular organisms. But meiosis is for sexual reproduction. So listen to the word, meiosis, sexual reproduction. And when we are going to reproduce, we must make sure that half comes from the mom, half comes from the dad, and we put the two together to make a whole you. So 50% of the chromosomes come from the mother, 50% from the father, and we join them to make you the new individual. That's why the cell, the genetic material must halve first, so that we have two halves equal a whole. Whereas with mitosis, the whole makes a duplicate of itself to make two wholes. And if you understand that concept, you cannot confuse the two. So mitosis, you're growing, making a brand new cell that looks like the previous one. Meiosis, you must halve the contents of the nucleus, so that when we put the two together, the two gametes, we end up with something that is a combined whole, which is a diploid organism. So haploid is half, diploid is together. And these are the terms you must know. They're in your X sheet, all those definitions. Make sure you go through them. It's important, because when a part of your exam paper is going to be provide the biological term for, and you must know it. All right, and it also helps you within your content. So let's look at cell division. We're looking at meiosis, but first we must look at mitosis. So mitosis, it's two identical daughter cells result when the cells are damaged. The surrounding cells will duplicate by mitosis to form new identical cells. Mitosis takes place for growth, for repair of the damaged cells, for replacement of those cells that cannot be repaired, and then reproduction in single-celled organisms. People, you must know that because they can and will ask it to you. All right, people, now we're moving on to the process of meiosis. Now, there are two main processes. You're going to have the first meiotic division, or meiosis 1, and you have the second meiotic division, which is meiosis 2. Now, how would you know which one they're talking about? Meiosis 1 would always have a 1 behind interphase, prophase, metaphase, anaphase, and telophase. IPMAT. Interphase, prophase, metaphase, anaphase, telophase. So it would always have a 1. And if it is meiosis 2, or the second meiotic division, it never has interphase. So it will go to prophase 2, and then IPMAT, so metaphase, anaphase, and telophase 2. And that tells you it's the second part of the meiotic division. All right, let's have a look. There are two steps of the meiosis process. Number 1, you have meiosis 1. And that's your first meiotic division. And this is called reduction division. Why reduction division? Because you are halving the chromosomes. So you're going to end up with the nucleus is halved. We have half the chromosome number. It's half. The result is 2 gametes that are haploid. Remember, if it is half the chromosome number, then it's going to be haploid. When it is the normal chromosome number, so it is whole, then we say the organism is diploid. In other words, it is a diploid chromosome number. So please remember, haploid, halving it. Diploid is so that when we join the two halves together, we have a diploid zygote. Now, all of this relates to when you get to reproduction. And this is why, for the new national curriculum statement, the meiosis portion was put into grade 12. So that relates to DNA replication and protein synthesis and nucleic acids. You then follow on to meiosis and then to sexual reproduction so that it all makes sense. All right, now, your second meiotic division here, you're going to have, so let's just underline it. This process is similar to mitosis. The haploid gametes are duplicated, and that is what is important. We are going to duplicate. Where before we halved it, now we're going to double those halves. And so in males, we're going to end up with 4 sperm cells. In females, we end up with 4 structures. Each of them are haploid, but one will develop into the egg, and the other 3 will form or provide nutrition for that egg cell. Now, the egg cell is the largest cell that we have in nature. It's the largest cell, and it's the size of, if you have an aerial fontaine and you have one full stop, that's the size of an egg cell. So it is quite large, generally speaking, compared to other cells. You can't see any other cells, but you can see an egg cell. You won't be able to see the detail, but you'll be able to see the overall structure. All right, so we're going to have, in males, you're going to end up with your sperm cells, and in females, 1 gamete forms the egg cell, and the other 3 remaining gametes form nutrition for this egg cell. All righty, so we got 2 phases. Now look here. Here it says prophase, what? Prophase 1. That tells you it is meiosis 1, or the first meiotic division. Here we have our normal cell, and what you do when we look at meiosis and mitosis is literally the nucleus takes over the entire cell. So here we have a chiasmata. It's portions of the genes of the daughter chromosomes cross over, and this is your biggest difference between meiosis and mitosis. With mitosis, you literally take that nucleus and it doubles up on itself. Whereas in meiosis, in your prophase 1, interphase, prophase, metaphase, meta is always middle, and tele is the end. So we have prophase, metaphase, anaphase. Your chromosomes are always moving towards the poles, always. And then you have telophase, where you have the dividing and the invagination that causes the two separate cells. So the chiasmata, very important. It is the crossover. Now, it is not crossing over or crosses as you have in genetics. So here we're not talking about a cross, saying male, this male and that female, and we're crossing them. This is called crossing over. So here we've got the two chromosomes. There they are. And what happens is the one chromosome interlinks over the other one, and genetic information is moved across, so that I'm now going to have this half of my finger will be from this one, and this half of my finger will be on that one, so that we have genetic variation. And that's exceptionally important for when we get to genetics. So this is where your genetic variation comes in. Otherwise, everyone would either look like their mother or look like their father, depending on which characteristics were the strongest. But we don't. They'll say to you, you look just like your great uncle Fred or your great aunt Madeleine. And you think, whoa, I don't look like that. And they say, yes, your jaw is the same as your great-grandfather or your great-grandmother, or you inherited your father's crooked, your great-grandfather's crooked pinkies. It's because of this crossing over which creates variation. All right, now, each chromosome has split into two daughter chromatids held together by centromeres. You see this? There you've got your two daughter, your chromatids, and they are joined here by a centromere. And this would be a homologous chromosome. Why homologous? Because homo means the same. So they are chromosomes for the same characteristic. This one would have come from your mother, and this one will come from your father. So what we've got here is this crossing over. Then we have metaphase, and when we look at metaphase, you're going to end up with your centrioles, remember, very important, and we have centrioles in animal cells, not plant cells. So the centriole is at the pole, and then coming off these centrioles, you have your spindle fibers, and your spindle or chromosomal spindle fibers will have chromosomes on, and your continuous spindle fibers don't attach to a chromosome. Something else, people, if they ask you to ever draw a phase in meiosis, your chromosomes are always, at this point, this even. They're always in pairs. You never ever have, they're always in twos, multiples of twos. You can never have an uneven number. All right, so if you start off with 4 chromosomes, you're going to end up with 2 chromosomes at the top and 2 at the bottom. So you always have it working in pairs, never an uneven number. So continuous spindle fibers, and then your chromosomal spindle fibers will have a chromosome on it. Then we look at the homologous pair. Homo, remember, means the same. Got a bit of frog in the throat there. Pair of chromosomes aligned at the equator. Now metaphase, remember, is the middle phase. All your chromosomes are lined up in the middle. Meta means middle. All right, so in the middle, and you have the number, the divisions of the centromere is going to be there now. Okay, so then we have the equator. The equator line should actually have been over here. It's in the middle. All right, now we move on to the next phase. Remember, prophase, metaphase. After metaphase, as those chromosomes start to move away, so early anaphase, that is when the nucleus starts to divide. All right, and when the nucleus divides, I'm going to write it on here, the nucleus separates. This is when we say karyokinesis. Karyokinesis occurs. Anything to do with the nucleus is karyo, and kinesis, to move. So that is when the actual nucleus starts to split, is directly after metaphase, when those chromosomes start to move away from the equator. So they move up towards the poles. All right, so your homologous pairs of chromosomes with one chromosome of each moving towards opposite poles. They have halved. This was a chromosome pair, and that was a chromosome pair, and they've now separated out. Now remember, we can't do this for a human cell, because if we did, we would have to draw 23 and 23, so 46 chromosomes, because there are 23 pairs of chromosomes, and we would just have one big mess. So we do this to explain it to you using only 2 pairs of chromosomes. The spindles are attached to the centromere, and it pulls those chromosomes towards the poles. So the difference between prophase, your chromosomes will be scattered around. In metaphase, they're all lined up in the middle. And anaphase, they start moving to the poles. And the reason I'm stressing this is because they can, in an exam, give you a number of diagrams and say, take those diagrams and put them in order. So you have to say, okay, if there's A, B, C, D, E, you have to say we have B first, then we have D, then we have E, then we have B, then we have A. And you need to put them in an order. So you must remember this. Prophase, everything is scattered around, the chromosomes. Metaphase, the chromosomes are lined in the middle of the cell at the equator. Anaphase, they pull in pairs. They start moving towards the poles. And then telophase. Now, some textbooks will write this as T-E-L-E, telophase, and others say telophase. Both are correct. One is an American version and one is an English version, and we all know that Americans don't really speak English. Okay, so as South Africans, we tend to go towards the British and towards the Oxford Dictionary version, and that would be telophase, but they are both correct. All right, so we now have a double-stranded chromosome. There we go, and it's arriving at the pole, and here we have the same happening at this end. The cell membrane now starts to constrict. They may say it forms an invagination. And that's quite a long word, especially if English is your second or third language. So if you just say that the membrane starts to constrict, in other words, it starts to fold in. The spindles begin to disintegrate because we don't need them anymore. They were there to pull the chromosomes to the poles. Okay, and here we're going to sit now with half of the chromosomes that we started off with, and in this side we have half. But now, look here. And this is very, very important. Here, what have we got? We've got this little area here where we've got the part that came from the father's or the paternal chromosome. And here we have the part that came from the mom. And the two are now sort of attached to each other, and they're working together. So this chromosome, even though it's the mother's chromosome, has a portion of the father's chromosome, and this one is the father's chromosome, it has a portion of the mother's chromosome. And that's what happened at the crossing over when prophase took place. So that's where your crossing over occurs, and it's very important because it adds to genetic variation. All right, now look at this. What have we got there? 2. And that 2 immediately tells us this is prophase 2. So I'm going to highlight this. We're going to take our magic marker. It is prophase 2. So it's the second meiotic division that we are now looking at here. So we look at our spindles. Okay, they form at right angles. So just after telophase has taken place, telophase 1, prophase 2 now starts. So we have the spindles form to the right, at right angles to that of meiosis 1. So if meiosis 1 was here, we now have meiosis 2 is at right angles. And there are our daughter chromatids, and they're very happy. Okay, here also remember the cell is haploid. Why? We have halved the chromosome number. Metaphase 2, meta is the middle phase. We've gone through that. And the 2 immediately tells us it's part of the second meiotic division. So here again, in metaphase, the chromosomes are lined up, whereas if we look here, they are scattered around within the cell. They're not organized. Here, They are completely, during metaphase, they're completely organized. They're lining up at the equator. There's your centromere, and there are the two chromatids. And we have our chromosomal spindle fibers, and we have our continuous spindle fibers. Centromere sitting at the top of the pole. All right, now, this here is anaphase 2. So it's happening during the second meiotic division. The daughter chromosomes are pulled towards the poles. Now look at what's happened here. They have duplicated. Okay? So in duplicating, what have we got? We now have our half, and the half has now duplicated. So we look at what we end up with. 4 daughter chromosomes that are unidentical. They are not identical, and I'll show you why in a minute. Let's just first look at this diagram. Telophase, clearly we'd have our constriction, and eventually the constriction, the cytoplasm and the cell membrane would form for each, and they would separate out. So here we have 4 cells with half the number of chromosomes that the original parent cells had, and each cell has a different gene combination. And this is exceptionally important, people, especially when we get to genetics. You are going to have 4 sperm cells. Of those sperm cells, you're going to have 2 cells. 2 of them are going to have X chromosomes, and 2 of them are going to have Y chromosomes. All right? And there you have, that's why it's the male that determines the number of, at least whether the baby is male or whether the baby is female. Because females only have X chromosomes. So we have a combination, we have X chromosomes in the one and we have X chromosomes in the other. But because of the crossing over at anaphase 1, you now have a genetic combination that is different. You are always, after meiosis, going to have 4 unidentical daughter cells, 4. Whereas at the end of mitosis, you have 2 identical daughter cells. Whereas here, 4 unidentical daughter cells. Now, let's see whether you've got this. For question 1, in fact, before I do this, I'm just going to go back here and just extend this page a little bit. Just want to show you something here. Okay, look at this. Here we have a mother, or the parent, female parent, and here we have the father, and I'm using mother and father, male parent, female parent. In the mother, we are going to have 46 chromosomes. In the father, we have 46 chromosomes, or 23 pairs. And here we have 23 pairs. Because remember, they're always in pairs, never an uneven amount. If there is, we have a problem. And then you end up with Down syndrome babies or Turner syndrome babies. We're going to work through that later as well. But let's look here. So now, we want to reproduce. It's not the mother's body fixing cells and repairing and replacing cells, or the father's body doing that. We want to reproduce. We have to produce in the father, we are going to produce sperm cells. And in the mother, we are going to produce egg cells. So we need to make sure that we're covering both. And this happens in the mother's body and in the father's body, or the parent female and the parent male. So if I want to add 23 and 23 together, so 23 pairs plus 23 pairs, I'm going to end up with 46 pairs, and that's going to be something very strange. It's going to be like an alien. We can't have that. So we say, no, no, no. The body is incredibly intelligent, and it fixes all of this. It says, that can't happen. That can't be. We need to halve. So in the mother's body, we're going to have meiosis taking place in the sex parts of the body. So in the female, it would be in the ovaries, or if it was a plant, In the plant, it would be in the ovary of the flower. In the male, we would have it in the testes of animals and humans. And in the plant, we would have it happening, on the stamens. The end of each stamen, you're going to have an anther. And that anther is where your male cells or your male gametes are produced. So here we're going to have meiosis as well. So meiosis is occurring in the male and in the female pretty much at the same time, according to this one that we're using here now. So in the female, we're going to take those 46 chromosomes or 23 pairs, and we're going to have reduction, division. So we have our 46 here, and we have reduction division, where we are going to halve the chromosome number. Something else, the mother is going to be written as 2N, which means she is diploid. And the father is 2N, which means he is diploid. You put this in brackets as well. Okay, here, 46 chromosomes, you're going to have reduction division. It divides into two. So, or it's halved, 2 parts. So we're going to have 23 chromosomes. And here we're going to have 23 chromosomes. And here we have 23, and I know I'm repeating myself, but I need for you to see this, chromosomes. And here we have 23 chromosomes. Now, this here is going to form a gamete. I'm just going to write a G here, and a G here, and a G here. Now, if it has 23 chromosomes, it is half of 46, so reduction division has occurred. And this is then going to be meiosis 1. That's where reduction division takes place. So we end up with 4 gametes. And these gametes must now be doubled up because we want to make sure that we have everything so that reproduction can occur. Remember, in a human male, a sperm count of between 90 and 120 million sperm per ejaculation is normal. I mean, for heaven's sake, we only need one of those little sperm cells to get to an egg cell for fertilization to occur. But the body wants to make sure that fertilization will occur. So it produces 90 million little sperm cells. If you look at flowers, an anther, many of you will be allergic to pollen. The pollen is what carries the 2 male gametes in it. And we're going to do plant reproduction as well. So that's coming. But you've got lots of pollen floating around, and each little pollen grain is going to have your male gametes in it. And it makes plants make millions of little pollen grains that go floating around so that the stigma of a plant or a flower can be fertilized. And the egg cell just sits. The egg cell doesn't have to travel anywhere. The egg cell just sits there waiting to be fertilized. So we only need 1 egg cell. We need plenty of sperm cells or the male gametes. So going back to our diagram here, we've got our 4 gametes. But now we want to make sure that there are plenty. So how do we do that? We say, okay, we're going to take these gametes and we are now going to duplicate. We are duplicating this whole process. So how do we duplicate this whole process? Let's go back to our blue, which is a nice. We're going to say, right, this gamete here, we are going to duplicate, and we're going to duplicate this one, and this one, and this one. Are they going to be the same? No, because they're going to have different characteristics. So if we go back here and we say, this was the male's going to produce sperm, even if we just take the sex cells, which are going to be, if these are your X and those are Y. They contain the X chromosome, those contain the Y. This is going to be X duplicated, and that's going to be Y duplicated. In the female, clearly they're all going to be X. So in the female, one of these will become the egg cell or the female gamete. The female gamete. And here we're going to have 2 X sperm cells or male gametes. And here we're going to have our Y sperm cells or male gametes. And all of these are haploid. So when I take this whole process and I say, okay, we want reproduction to take place, we are now going to take a haploid egg cell and it is haploid. In other words, it contains half the chromosomal number of the female. And during reproduction, we have our little sperm cell or our male gamete. And the 2 of them fuse. Let's use the 2 of them fuse. All right. What is that process going to be called? It is called fertilization. And once fertilization has occurred, we say, after that, what do we have? We are going to have, let's use black, we are going to have a 2N, which is called diploid zygote. So quite literally, N plus N is equal to 2N. And all of you can count to 2, can't you? So N plus N gives us 2N. A haploid sperm cell, a haploid egg cell, fertilization, and we now end up with a diploid zygote. And that zygote will now undergo mitosis. And why? So that it can grow and mature. And when it is an adult, it will itself undergo meiosis in the sex cells, in the female meiosis in the sex cells, so that we halve the chromosome content to be haploid, so that the 2 haploid gametes join to form a diploid zygote. And that is the cycle of life. Whether it occurs in the plant kingdom or the animal kingdom, that is how it works. And this forms the basis for reproduction, which is an entire module all on its own. And they are quite emphatic, your examiners, that you must understand the basic concepts. And that is why meiosis is incredibly important. So I hope this now makes sense. So let's go to our questions. We have, and this is what they're going to give you in your exam paper. All right? They say, okay, here we have A, we have B, we have C, D, E, and F. First thing you are going to do is label your diagram. So we've got our chromosome with 1 chromatid, the centromere. We've got the chromosomes with 2 chromatids. Here we have our threads, and in C, they are lining up in the middle, which is metaphase. Then here they're pulling towards the poles, and here we have the whole process taking place where we end up with 4 chromosomes. So what they've done is they've abbreviated and made the whole process very, very simple. Now, if we look here, This phase here would be interphase. This phase here, where we've got our chromosomes with 2 chromatids and our chromosomes are in homologous pairs, this here would be prophase. Then we would have metaphase and anaphase. And this would be early anaphase. This would be late anaphase. How do we know? Early, well, it would start here where the chromosomes were here, and the further they travel towards the centromere, the later the anaphase stage. So here they've pulled completely away. And here, what have we got? They've already split into 4, so this would be late, if we really want to get technical, late telophase. Telo, fine. All right, now let's look at the questions. How many chromosomes does cell B have? So people, you go back to your diagram and let's see. How many chromosomes does it have? It has 4. 1, 2, 3, 4. If they had said to you how many chromosome pairs do we have, then what would you answer? You would answer 2 pairs. Remember, if I have socks, we don't only have 1 leg, we've got 2, generally. So, your 2 feet, you need 2 pairs, you need 2 socks. That makes 1 pair of socks. So how many chromosomes does cell B have? It has 4 chromosomes, not 2 pairs. It's not asking you. It's asking you how many, not how many pairs. So just how many chromosomes. Then, would this be a haploid or diploid? Give a reason for your answer. So if we look up there, has it divided yet? Have these chromosomes separated? Because remember, this will be your paternal chromosome, that will be your maternal chromosome, your maternal chromosome, your paternal chromosome. So, has it separated out yet? No. Therefore, it is still in a diploid state. So, would it be haploid or diploid? It would be diploid. It would be 2N. And we can say there are 2 sets of chromosomes. Or you could say your homologous pairs. Remember, homologous pairs of chromosomes are present. Okay, and they actually tell you here they are homologous chromosomes. Let's just move up here. Pair of homologous chromosomes. So it's there. You must just look you must know where to look. All right? And if you know this anyway, it's there. So you don't have to make spelling errors. You simply just copying it down. But it is definitely diploid. At this point here, it is haploid. But there, it's still diploid. Okay, people, now we're going to look at question 2. And this will give you another variation of what they can ask you, or the type of question they can ask you. So this was taken from the National Senior Certificate. That's what NSC stands for. Life Science Exam, and we're looking at February 2009. So this was last year's supplementary paper. And it's going to be very similar to what the learners got for the October, November, December 2008 paper. Your supplementary papers are always very similar to your final papers, and the reason for that is they expected you to know a certain amount of content in November, and if you fluff that exam, you have a second opportunity to rewrite it when you do your sub-papers. All right? So this is February 2009. And if we look at the first rule is look at the diagram before you even look at the questions. Look at the diagram and label anything that is required on that diagram. So, number 1 would be a chromosome. And you do this on your paper, all right? These 2 here would be a homologous chromosome pair. Now, just to refresh your memories, homologous chromosome pair means homologous, the same, so to be for the same characteristics. Let's say you have a chromosome that has from your mother that determines eye color and hair color and skin color. Then from your father, or the paternal side, remember maternal is mama, paternal is papa, and that's how you remember it. So maternal, that we're looking at eye color, hair color, skin color. And for the dad, you're looking at eye color, hair color, skin color. Those chromosomes are carrying characteristics for the same traits. So we're looking at hair color, eye color, skin color, daddy's one, mommy's one, and that would be a homologous chromosome. So that's a homologous chromosome pair, and this would be one of those chromosomes. Now, if we look at B, B's arrow should be here. So we're going to do an arrow and make it to where it was supposed to go. Apologies for that. So we're not all perfect, but we can try. We strive for that. All right, so B is your centromere. And your centromere is the part that joins your two chromatids together, which is what label C is, chromatid. And your chromatid sits there, and you've got one chromatid and another chromatid, and when it joins in the middle, that's your centromere. And that's why it looks like a shape of an H. All right, and then D, this process here that's happening there is called the crossing over. And it's also called the chiasmata. And chiasmata is a fancy way of saying crossing over. So if you can't remember chiasmata, crossing over is fine. And what happens in crossing over? We get the genetic information from the maternal chromosome crossing over with the paternal chromosome from the father, and they start to share information. All right, so just, I just want to just make sure that you understand this very carefully and very clearly. Look here. In crossing over, there is the father. So this is the paternal chromosome. Okay, we can do the mommy one in purple. So this here is the maternal chromosome. All right, joined in the middle by a centromere and joined in the middle here by a centromere. So chromatid, 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 centromere, centromere. Okay, during crossing over, which happens during prophase, remember interphase, prophase, metaphase, everything's in the middle, anaphase, it's pulling towards the poles, and telophase, we end up with our separate cells. Okay, so here, what do we have? We have our two chromosomes. These two will make one homologous chromosome pair. Okay, so they sort of drift together, and I'm going to move the mother one. So this one now moves. And we end up with the crossing over. So what happens is a piece of the mother's chromosome goes across to the father's chromosome, and a piece of the father's chromosome now goes onto the mother's chromosome. So our result and it won't move down. So I'm going to just do this. The result is going to be the paternal chromosome and the maternal chromosome. I I keep wanting to use my finger. This is very, very funny, actually. It's just... And that's what you're going to sit with. So your homologous chromosomes now, the paternal chromosome is going to be carrying information from the mother chromosome or maternal chromosome, and the maternal chromosome is going to be carrying information from the paternal chromosome. So when they end up dividing to form your haploid gametes, you are going to have this information with a piece of the maternal, and this information with a piece of the paternal. And it would depend on which combination ends up going in to make the diploid zygote. All right, so let's just take this up again. And we say, right, first thing that you have to do is answer to provide the labels for parts A, B, C, D. We've already done that. We've said A is the chromosome, B is going to be the centromere, C is the chromatid, and D is going to be the crossing over or chiasma. And people, even though the answers are there and you've written them on your question paper with your diagrams, and you may sort of think, oh, this is a waste of time. It's not. It isn't ever a waste of time, because not always do they ask you for all the labels. They may only ask you for one label, but to explain the process that's occurring, you need to do this as part of exam technique. All right, then, name the process in meiosis that is illustrated in the diagram above. Now, the process going on, chiasma only, or the crossing over, only ever takes place during prophase. Now, remember, we have the first meiotic division and the second meiotic division. It's going to be prophase 1. Because that's when reduction division takes place. And reduction division only takes place after the chiasma has occurred. Okay? State one importance of the process named in question 2. It is for genetic variation. Okay? So that you can have your great-great-grandfather's nose and your great-great-great-great-grandmother's crooked pinkies, or your great-great-uncle's funny legs and funny-shaped knees. That is life. That's what makes us so unique, is we don't just look like our mothers and our fathers. We look like a combination of our moms and our dads, and our uncles and our aunts and our grandparents and our great-grandparents and our great-great-great-grandparents. And that's what makes us wonderfully unique, because you've got little bits of all of them that make us as we are. During which phase of meiosis does this process named in question 2 occur? What have I done here? Which phase would be prophase 1? And I've just cocked up. I'm sorry. Fuck. Okay, people, now you've got that on your paper. Let's have a look. 2.2, name the process in meiosis that is illustrated in the diagram above. So that's very easy. What are we looking at here? We're looking at this, which is the chiasma, or the crossing over. So now, don't try and be clever in an exam and write both. You choose whichever one suits you, and that's the term, because they mean the same thing. All right, so it's the process in meiosis that's illustrated. Then it says state one importance of this process. Well, guess what? The process is going to be genetic variation. And that is so that you can look like your great-grandfather, and you can have your great-great-great-aunt funny fingers, or funny little tip on your nose, or dimples. Those are all family traits, and you are a combination of all the family traits that have gone before you. So isn't that wonderful? That's why we're so special as human beings. During which phase of meiosis does this process? If you do not know this by now, I'm going to hop through here and I'm going to give you a shake. All right? And if you don't, give that head of yours a shake. It is going to be prophase 1. So it's always prophase 1, that indicates that it occurred in the first meiotic division. So meiosis 1. Then draw, now here it starts to get a little difficult, but it's only for 2 marks. And it says here, draw a diagram of the structure labeled A to show its appearance immediately after the process. So they want you to take this diagram A, in other words, our chromosome. They want you to draw that chromosome, this one, after the process has taken place. So how do we do that? It is going to look, A is going to look like this. So there is diagram A. And that's all they want you to see, want you to show them. You are going to label it chromatid, at least chromatids, centromere, and you're going to put portion from the other chromosome. And that's it. They want to see that you know that that's what happens during the crossing over. Simple little 2-mark question. Okay, now we move on. What is the significance of meiosis? Why is it important? Well, guess what? If it wasn't for meiosis, we would not be able to reproduce unless we were single-celled organisms, and we are not. We've progressed. We have, to use the term, evolved. And evolution, or the word to evolve, means to change over a period of time. So, in fact, it's a very long period of time. So, formation of haploid, now we're going to use our magic. They are haploid gametes, all right? And those haploid gametes form from diploid chromosomes in preparation for what? For sexual reproduction, people. Sexual reproduction. So meiosis is for sexual reproduction. The formation of haploid sperm cells during meiosis is called spermatogenesis. Now look at the word. Sperm, you are producing sperm. And genesis means to create. That's why we have the book of Genesis. It means to create. So it is to create sperm cells. Now in females, we're not making sperm, we're making egg cells. And in life science, or the biological term for it, we're going to talk about ooh, means to make the eggs. Now, I mean, an egg is round, so it's not hard to remember. So we have oogenesis. And these 2 terms are incredibly important for when we get to sexual reproduction. And I know I've said that a number of times, but this is why we do meiosis, so that you have a grounding for when we go to sexual reproduction. So we want the formation of haploid gametes from diploid chromosomes. And in males producing sperm, which are haploid, we're going to have spermatogenesis. In females, those haploid eggs are going to be by a process of oogenesis. And then we also ensure that the chromosome number remains the same after the offspring, in the offspring as in the adult. So we're going to have a haploid plus a haploid gives us a diploid. So this came from father 1, this came from mother 1, and that gives us you and me and every single organism that is reproduced, every offspring. That little creation, that diploid zygote that grows by mitosis into a adult organism, whether it's an adult bunny rabbit or whether it's an adult dog or whether it's an adult cat or whether it's an adult you. That is the process. So haploid gives you diploid. Ensures the genetic variation occurs when crossing over takes place in prophase 1. People, please make sure that you understand all of this. All right, now let's see how on the ball you are. In the production of sex cells, in plants, so this is what we're looking at in plants, the spore mother cells, okay, in the pollen sacs, so it's in the pollen sacs of the anthers, are going to divide by what process? By meiosis, all right? And when they reproduce by meiosis, those pollen grains are going to contain 2 male gametes. 2 male gametes, all right? Why? Because in plants, we have what's called double fertilization. And double fertilization is the one little male gamete joins with a female gamete in the egg cell to produce what? To produce the new little organism, the diploid zygote, which will then be inside. That embryo will be inside the seed. But what makes the fruit? Now, you eat apples and you eat bananas and pineapples and plums and peaches. The fruit that you are eating is actually the ovary. And that ovary that's grown into a nutritional base for that little embryo that's growing inside the seed. So there you go. First of all, you're eating ovaries when you eat an apple and a pear and a banana and a peach. Okay. Secondly, it is as a result of the second fertilization in the double fertilization of plants. All right. So we are going to have the spore mother cells. We're going to have the anthers meiosis to produce what? The pollen grains, and they are going to contain 2 male gametes for double fertilization. A specialized cell in the ovule divides by meiosis to produce the embryo sac, and inside that embryo sac we are going to have our little egg cell. And that egg cell, your male gametes are going to be haploid, your egg cell is haploid. In animals, what do we have? Your male gametes or sperm cells are produced in the testes. In the female, your egg cells or female gametes are produced in the ovaries. Now people, quite important here, the 23 pairs of chromosomes in humans result that we have a diploid zygote. So it divides as follows. 22 pairs of autosomes, which are your somatic cells. They result in your somatic cells or your body cells. Okay? And then we have 1 pair of sex chromosomes, and we've already said this. In the male, we have XY, and in the females, we have XX. All right? Quite important. You must know that. All right, people, now we have to look at the comparison between meiosis and mitosis. And if my gut feeling is anything to go by, you will be asked some form of comparison between meiosis and mitosis in an exam, either prelims or your final exam. So make sure you know it. Also, it's good for you to understand, and if you do understand this and know it, then it makes sense of everything. All right, so if we look at mitosis, remember, mitosis means that you have growth, repair, replacement of cells. So it's always, where does it occur? In the somatic cells, which is your body cells. Let me just do this so that you remember. It is every single body cell that you have. All the cells excluding your sex cells. Meiosis is for sexual reproduction. So therefore, it's going to occur in the reproductive organs. So in plants, you're looking at the ovaries and the anthers, or the stamens at least, and anthers, end of the stamen, and then you have the animals, the ovaries, and the testes. Okay, now why the process occurs? Why does it happen? Well, first of all, because mitosis is to make other new cells that are identical, you're going to have genetically identical cells. Why? For growth, for repair, and for replacement of cells. And then for reproduction in unicellular organisms. Why? Because they only have one cell. There is no reproductive organ. So if there is no reproductive organ, guess what? You have that one little cell that then undergoes mitosis to make more of itself, like, for example, bacteria. Okay, in meiosis,
Well, guess what? It's for sexual reproduction. So why does this process occur? To reduce the number of chromosomes. So we have reduction division, so we end up with a haploid gamete. Now, how does this work? Well, your haploid gamete, let me just show you here. We go from a diploid nucleus, which then reduces into 2 haploid gametes. Now, we'll have this happening in the male. We'll have the same thing happening in the female. Now, during fertilization, what are we going to have? We're going to have the N from the male. We're going to have the N from the female, okay? And that's going to join to go back to 2N. So just let's have a look here at this whole process. So it's reduction division resulting in haploid gametes. Why? For sexual reproduction. Okay, so that your chromosome number remains the same when you have the offspring. So we're going to have haploid gamete from mommy, haploid gamete from daddy, and we're going to end up with our diploid new little structure, our little offspring. Similarities. Now this is incredibly important because this is where learners get confused. With your similarities, you've got transformation of the chromatin network to chromosomes. You have karyokinesis takes place. Remember, that's the splitting of the nucleus. Nucleus splits. That's karyokinesis. Cytokinesis is when the cytoplasm splits. All right. Now, if we look at meiosis, you have a transformation of the chromatin network to the chromosomes. It's identical. You're going to have karyokinesis takes place, and you're going to have cytokinesis taking place. So that's easy because those are the things that are the same in both processes. But let's look at the differences. And the differences is what makes mitosis and meiosis so special. In mitosis, you only have 1 nuclear division, whereas in meiosis, there are 2. You have reduction division, and then you have duplication. There is no crossing over in mitosis. You don't need it. Why? Because you're not swapping genetic material. It's not for variation. It's for, let me think. If for example, I have my lips are very dry and my lip splits and I get a bit of a sore there and it's sore, it hurts. New cells, I don't want heart cells or lung cells growing on my lip. I need lip cells to come back. If, for example, you have a pimple and it gets all red and sore, and when it heals, what do you have? You have new little cells that are identical to the other skin cells. If I lose a heart cell or a lung cell or a kidney cell, it must be replaced by the cells that are the same. So that is why we only have 1 nuclear division and there is no crossing over. Why? Because there is no need for variation. There's no need for variation. Whereas with meiosis, we have 2 nuclear divisions. One is to reduce and then to duplicate. So here we're going to have 2N, then becomes N, and the N's then duplicate, so we've got lots of them. And all you have to do is think of the number of sperm that are produced, or male gametes. Now, crossing over is essential. Why? Here we didn't need variation, but for meiosis, we definitely do. We want genetic information that is exchanged. Why? Because we want variation. And all you have to do is look at your brothers and sisters. Do you look identical, like identical twins to your brothers and sisters? No, you don't. You are definitely from your mom and your dad, but if you have 5, if there's 6 children, every one of you will look like brothers and sisters, but you'll all be slightly different. That is people, that is variation. And you don't look like your great-grandmother who looks like your grandmother, who looks like your mother, who looks like you. Otherwise, all the women on this earth would all look the same, and all the men on this earth would all look the same. We don't. We have variation. Okay, moving on. again with differences. So these are more differences. The daughter chromosome is pulled to the opposite pole. All right, so it's the daughter chromosome is pulled to the opposite pole. Here, your daughter chromosomes don't separate. So we have one entire chromosome of the homologous pair is pulled to the pole, and each chromosome pair separates independently, and that also results in variation. Okay. Another difference we're going to have is we have two identical cells are produced. They are always two identical, and we call them two identical daughter cells, okay, with identical chromosomes to the original cell. If this didn't happen, we would have a huge problem because we would end up with heart cells on our cheek. Now, meiosis, we have 4 cells are produced, but look at this. Here they are identical. Here, they each contain half the chromosome number, all right, and they are different. So half the original chromosome number as to the first cell, first duplication that occurred, or reduction division that occurred at least. And so we have reduction division, and then we have duplication. And in that duplication, for example, with males, you're going to have X and Y chromosomes. So that will split up. All these differences that you're going to have, whereas with mitosis, we have 2 identical daughter cells. Meiosis, 4 unidentical structures. Okay, now we go to diseases and syndromes. Very, very, very complicated, but also uncomplicated and easy questions. If you know that and understand the basics, then everything's good. We have an issue with the chromosome numbers that may occur in meiosis. So, for example, we have 46 chromosomes in the male and 46 chromosomes in the female nucleus. In the male, it divides into 2 times 23, and the female's the same. But sometimes there's a little tiny problem. Something creeps in, and we end up with, in the female, 22 in one nucleus, or one daughter cell that results, and 24 in the other. And this is when there is a problem. So when either of these resulting gametes joins with a normal gamete, this is what happens. Now, and this is just basic maths. You have 23 plus that 22 gives you 45, when it should be 46. It should be 23 plus 23 chromosomes. 23 haploids from the mom, 23 haploids from the dad gives you 46 diploids in the zygote. Here we've got 23 and 22 gives you 45. There is a problem because we're missing a chromosome. So what do you think is going to happen? It's going to cause a severe mutation, or the body will spontaneously abort. Or if we have 23 plus 27, oh goodness gracious, now we end up with 47, and that is a problem because it's not 46. So that's when we have abnormalities, and those abnormalities are as a result of meiosis. So if we look at Down syndrome, there are 3 syndromes you need to know. Number 1, Down syndrome. And here the baby has 47 chromosomes. So the egg cell has 24 instead of 23 chromosomes. And then we add the 23 sperm cells, and that's where the problem comes. And it's normally mothers or women that are over the age of 40. So as you head towards after 40, the chances are that your egg cells are not going to have 23 chromosomes in them. They may have 24. And then that joins with a sperm cell of 23, we now have 47. And that's why the chances of having a Down syndrome baby increases from the age of 35. And after the age of 40, you almost have a geometric increase. All right? We have 45 autosomes, and then there are 3 number 21 chromosomes. Because remember, the 23 pairs of chromosomes are all numbered. So chromosome number 21, we are going to have 3 of them instead of 2. And then we have a problem, and then we have 1 pair of sex chromosomes. So the characteristics of a Down syndrome baby are, and if you go do yourself a favor, go to the media center, have a look in books for pictures of Down syndrome babies, go onto the internet. There's a lot about Down syndrome babies, and you'll see the Down syndrome children have very much a very specific physical look about them. So first of all, you're going to see it's obvious at birth. You have the head, the face, and the body have a characteristic shape. Then you also have congenital heart defects. Very, very common with Down syndrome babies. And also, this is the big one, there's mental retardation. Now, clearly, some Down syndrome children are have a greater mental retardation than others, but they certainly do not function like a child who has 46 chromosomes in their nucleus. So there is mental retardation, and the children very rarely develop after the age of, they've developed mentally till about the age of 5. And then it gets sort of sometimes 6 or 7, but it's mainly 5 years of age. They're very sweet, loving, wonderful, wonderful children, but they remain children for their lives because they cannot develop and mature like normal people do. So that's your Down syndrome. It can be detected during pregnancy. That's quite important. The mother has something called an amniocentesis test done. And there they would take a syringe and they draw amniotic fluid because it'll be full of cells that have come off the baby's body. And they then check those cells and they can check. So it's only if you suspect, which is generally if you are over 40 and you're pregnant, you are going to go and check it. This procedure, however, can only be conducted during the 6th month of pregnancy. Now, people, you will know legally, girls can, or females can have an abortion up to the age of 3 months. So pregnancy up to 3 months. Between 3 and 6 months, you're in your second trimester, I mean 3 mesters, So babies develop over 3 trimesters. You can abort freely in the first trimester. The second trimester, it starts to get dodgy, but the limit is 6 months. It is only for Down syndrome and really severe genetic problems that they will approve and legally allow you to have an abortion after 6 months. But here it is clearly very much a parental thing, and it'll be up to the parents to undergo genetic counseling to tell them exactly what the processes are, what the problems are, and what that child would be like, and all the problems that would go with it. For example, a child with congenital heart disease is going to have to undergo heart surgeries, and it's not just the cost, it's the pain and suffering that the child has to go through, and also the siblings of that child and the parents. And listen, they can in an exam ask you questions like that because they want to know what your value systems are. Now, there isn't really a correct, a right or a wrong answer there, but make sure you can back it up with fact. All right? So, should the developing embryo be found to contain 47 chromosomes, it is up to the mother. The mother will have the first option to terminate, and she would then also clearly talk to the father. Now we look at biotechnology. Now, what is biotechnology? Now, use your common sense. You know, whenever there's a word or a term that you think, whoa, this is major, split it up. Bio would be biological, and technology, well, we all know about technology. You can type fast on your cell phones that most people can type on their computers. So, biotechnology is the use of microorganisms and also biological substances like enzymes to perform specific industrial or manufacturing processes in agriculture, food science, and medicine. Now, if we look at biotechnology, we start looking at genetically modified foods, etc., and everybody has a little cadenza about it. But it has a positive to it. So we need it. We need a lot of food in this world. And if we don't have that food, it's not going to work. So scientists use biotechnology to artificially select characteristics in plants and animals that suit the needs of humans. Because remember, animals can't do biotechnology. They don't have the intelligence to do it. It's us selfish human beings that inhabit this planet. Okay, the result can be classed as an evolutionary mechanism. Now, people, what is evolution? Evolution is simply change over time. Diversity is the result of that change over time. Evolution does not mean that you need to give up religion and now believe that we're all related to the chimpanzee in the zoo. We are talking about the word evolution simply means change over time. Diversity is the result of change over time. Okay, not hard. So what the scientists do is they pick out specific characteristics. Let's say, for example, Great Danes. They're huge dogs. So they are bred for their size and their height. So they would have looked at a whole variety of dogs and said, we want a dog that is big, that is high, stands high off the ground, has a really big body and looks like a calf. And they then select those specific genes and those characteristics, and they breed and they breed and they breed until they end up with a Great Dane that's a meter bigger than the normal Great Dane that we know. Or if you compare a Chihuahua and a Great Dane, there's a huge difference there. So the scientists specifically select characteristics from different organisms, and then they try to interbreed them. And it results in it's an evolutionary mechanism. Why? Because we end up with a hybrid. A hybrid is a mixture. That's all it is a mixture. So, and you end up with a new species. So we say it is genetically engineered. Now, it will have this new organism, this new species, will have new characteristics, so different characteristics from the original species. And now remember your gene frequency, and that results in DNA changes. So new characteristics, new gene frequency, and therefore DNA changes. Now, what do we end up with? In animals, we talk about new breeds. In microorganisms, you talk about new strains. Now, come on. You are forever hearing about a new strain of flu. And then everyone has to go off to the chemist and to their doctor to get inoculated for this bad flu that's attacking us this year. Every winter, we have 4 or 5 strains of flu virus that attack us. So when we talk about microorganisms, we refer to them as strains. Every different species will be a strain. With animals, we talk about new breeds. And then with plants, we talk about varieties. Okay, and they're created quickly and easily. Now, there's something else here that's very important. This hybrid or mixture that results when we have new breeds or strains or varieties, etc., sometimes it will have, that hybrid will have advantages. Now, what is it? We can, in agriculture, increase body size. So you get bigger sheep, bigger cows, bigger pigs, and we get more meat. Increased production of eggs, offspring, or seeds. Increased resistance to disease. Reach maturity quickly so that they can grow quicker so we can eat them quicker. Okay. And all of this is what we do as humans. Now, if we look at polyploidy, again, look at the word. Don't think, oh my gosh, I'll never learn this. Poly means many. Ploidy, well, you know, diploid, haploid, it refers to the chromosome number. Okay, the complete sets of chromosomes. Now, where all the cells have multiple pairs of chromosomes beyond the basic set. So, polyploidy refers to the change in the gene frequency, so that's where the genes are placed and the number of genes, and therefore the chromosome numbers, because that will change. And that is what alters the species and puts it at a different genetic level. Now, when chromosomes are different, your bivalence cannot be formed properly during meiosis, so gametes can't be produced. Remember, your bivalence is as they separate out and they move to the opposite poles. So if they aren't there, it can't work. Now, in animals, the polyploidy hybrid, like for example a mule, it's an example of a polyploidy hybrid, is when we take people, a donkey and a horse. You end up producing a mule. Now, a mule is incredibly strong. Okay, so it's got the characteristics, because remember, mules are actually very strong. Now you take the strength of a mule and the strength of a horse and you put them together. A horse is bigger than a donkey. So you end up with the strength and the bigness, the increase in physical size. But shame, they are sterile. They're infertile, they're sterile. So they cannot actually undergo sexual reproduction and produce more of its kind. Now, if we look here, it's common in plants, okay, and doesn't affect reproduction in plants. So it's plants is where polyploidy actually counts, all right? Sometimes we end up with sterile crops. Now, what we need to look at here is I love seedless grapes. Why? Because it means my son doesn't spit those pips out, and when I walk on my carpet, I can feel the odd pip here and there. So seedless grapes are amazing. Watermelon, seedless watermelon is lovely. Kiwi fruit, lovely, delicious. Now, all those different types of fruit are sterile. They can't reproduce. I mean, clearly, a watermelon that doesn't have pips, well, there are no seeds, therefore, you cannot grow a new watermelon. So sometimes we have different cultivars of many seedless types of fruit. Now, these crops are propagated, and clearly, if they have no seeds, you have to use asexual techniques. And people, your asexual techniques, grafting, cutting, stolons, you know this. It was in your grade 6, agrees to your grade 8 and 9 syllabus. So they are seedless. Therefore, there is no sexual reproduction. And if there's no sexual reproduction, guess what? There are no seeds. So therefore, we now have to look at asexual reproductive techniques. Polyploidy in crops can be induced by treating seeds with a chemical. Now, this chemical is actually very special. It's called colchicine. And colchicine inhibits the chromosome segregating during meiosis. So what do we end up with? We end up with, now they've got to move to a pole. So during metaphase, you've got all the chromosomes lined up at the equator, and this colchicine stops them from separating out so that they move to opposite poles. So we end up with all of them moving to one pole, either the bottom or the top pole. And when that happens, we end up with half the gametes have no chromosomes, and the other half will contain double the usual chromosome number. Because remember, you had duplication occurring. Now, the resulting embryos have double the usual number of chromosomes and can be tetraploid instead of diploid. So diploid would be 2N. Tetraploid, tetra means 4, so it would be 4N. It has double the usual. And this type of genetic manipulation in animal cells results in death. We can't do that in animal cells. Now, the result in plants is that they're bigger, they're tougher, they grow faster, and everything is hunky-dory and everyone makes more money, and we can feed more people. Now, what I've done is I've put in a whole bunch of examples here of different polyploid crops. And if we look at these polyploid crops, triploid would be 3N. Apples, we all love apples, bananas, watermelon, many types of citrus fruit. This is just normal. These are not the ones that end up being seedless. We get tetraploid crops, that's 4N. And we've got durum wheat. Come on, you all know pasta. Pasta's made of durum wheat. Maize, good old pup, mealie meal. We've got cotton, potatoes. What would we do without potatoes in this country? Okay, hexaploid, hexa means 6, so it's 6N. Here we have many varieties of flowering plants. Bread wheat. We all love bread, don't we? Kiwi fruit, very special. And then we have, now look at this one, octaploid. Oct means 8. So it's 8N crops, 8 sets of chromosomes. Strawberries, don't we all love strawberries? Sugar cane. Now, what would we do without sugar? Okay, and sugar cane is an octaploid. So in plants, people, polyploidy is natural. It's not a problem. It's when we look at doing animals and we start messing with the ploidy, There, it ends up in a fatality. We cannot have an organism. It just doesn't get born. It gets miscarried. All right, so it's as simple as that. Hope this makes sense, and just remember, polyploidy is only for plants and not animals. That's it for this session. Have an absolutely awesome time studying, and remember, the effort you put in now is the effort you're going to get out at the end of it. And we have one big exam to work towards, and that's the end of year 1. So have a good one. See you next time.